Claire Schon. So Claire Schon is a graduate student in the Waterloo Wetland Lab at the University of Waterloo with interests in botany, wetland ecology, and restoration. She's studying the biological control of Invasive Phragmites australis in southern Ontario. Her research is a part of a broader pilot program in North America for the biological control of invasive Phragmites. As a plant ecologist, she is interested in characterizing the impacts that the newly released stem-boring moths have on invasive Phragmites growth, physiology, and reproduction. Claire is also establishing long-term monitoring protocols for a wider plant community following biological control agent release. So with that, I will ask Claire to move on and put her screen up. Awesome. Thanks, Claire. It looks good.
Okay, thank you. So yes, hi everyone. I'm a graduate student in wetland ecology at the University of Waterloo, and I'm going to be talking about the biological control of invasive Phragmites australis. So if you've just watched Michael's talk, don't worry, you're not going to be seeing or hearing double. I'm going to be focusing on some of our plant responses in our first year of releases. So Michael's already introduced us to our two biological control agents, Lineasa gemnopuncta and Arcanara neurica. And while the entire life cycle of these insects are completed within patches of Phragmites, when we're focused on the damage to Phragmites, as we are in a biological control program, we're interested in this larval phase here. This is because as the larvae move through their larval development, they're boring into multiple stems. As they bore into the multiple stems, they're feeding on the inner stem tissue, and really what they're doing is damaging the vasculature of the plant. So on the left, I've drawn some of the plant responses that we see in the field, and Michael's started to describe them, but I've drawn an undamaged ramete for reference. Just so you know what I'm referring to, the ramete is the above ground portion of the stem, as Phragmites is a perennial plant and it lives year to year. And its underground network of roots and rhizomes, it sends up these aerial shoots every year, and that's what we call a ramete. The first response to this larval feeding is a dead ramete that essentially wilts completely and it loses all of its live tissue. It can't survive after the larval damage. The next response that we see in the field is this formation of secondary branching. So you'll notice that these upper leaves on the primary stem have wilted completely, but below where the moth had fed, we see this secondary branching start to form, and we also call these side shoots, but really what it's doing is trying to reroute its vasculature and compensate for some of the damage. So throughout the season, these branches or side shoots will produce more leaves, and they'll be able to keep growing for the plant. Lastly, we also see this like wilt only, just where the stem, the leaves at the top of the stem wilt, but the lower ones will remain alive, but the plant growth is stunted for the rest of the season. From research in Europe, we know that this larval feeding reduces the height, the above ground biomass, and the flowering of these rametes. And over time, we expect that these reduce the competitive ability of invasive Phragmites. So Phragmites really effectively outcompetes our native plant species right now. So we hope that these reductions that we're expecting to observe will allow for plant community recovery to occur over time. And like Michael said, there's moth releases across 13 sites in Ontario, and we work at a subset of those two sites. So we work both at the University of Waterloo and at Rare Charitable Research Reserve. And we, as part of an ongoing release experiment, we released these moths as eggs and as larvae in April and May of 2022. And from these releases, we were able to ask 3 plant-related questions. So the first question that I'll address is, how is ramete growth and reproduction impacted by the larval feeding from biological control agents? So to address this, we harvested individual rametes of 3 different kinds. We harvested the branching rametes, and if you'll see on the left here, there's this newly formed secondary branch. This is pretty early on in the season, but it will continue to grow throughout the season. We also harvested the dead rametes and as well as healthy rametes. And we harvested these in October after the kind of experiment or the growing season had completed. So we have these end of season results. And on each ramete, we measured the height, the stem biomass, the leaf biomass, the branch biomass, as well as the whole ramete. So adding up the different metrics of stem, leaf, as well as branching biomass for our branching rametes. And looking at some of the results of these, we have 3 different types of rametes represented by the different colors. So in our light green, we have our control rametes. In our dark green, we have the branching rametes. And in our orange or yellow color, we have the dead rametes. And the different metrics we're looking at are stem height, stem biomass, leaf biomass, as well as the whole ramete biomass. And so these are all in response to this early larval feeding. And we look at stem height, we find of the branching rametes a 42% reduction in height, 52% reduction, and with our dead rametes, a 52% reduction in height. So what this is telling us is that after larval feeding, these rametes are much shorter than the control or healthy rametes. With our stem biomass, we found a 52% reduction of the branching rametes and a 68% reduction of the dead rametes. So just on the stem alone, they're producing less biomass. With the leaves, we found, with the branching rametes, we found a 62% reduction, and a really stark contrast with our dead rametes, we found a 93% reduction of leaf biomass. And so especially with these dead ones, they're really stuck with whatever leaves they had produced early on in the season before larval feeding. So that could just be 1 or 2 leaves, but the branching ones are able to still produce leaves throughout the season, but not as many leaves as the control rametes. With our whole ramete biomass, when we're adding our different metrics of biomass, we find a 48% reduction with our branching rametes and a 75% reduction with our dead rametes relative to the control. So looking at this branching ramete here, or the results from the branching rametes, even though they're trying to compensate for the damage, they're not really doing so effectively. They're not able to keep pace with our healthy rametes. So over all of our metrics, we found that larval feeding consistently reduced ramete growth. With our next metric, we're looking at reproduction, so the formation of this panicle. And this is just one method of how Phragmites reproduces. I know we've already mentioned some of the vegetative modes of reproduction, but for rametes alone, we're just looking at the panicle production. And so this is just a count of the number of rametes that did or did not produce a panicle. And we find for our controls that 29 did produce a panicle and 2 did not. We find the exact opposite with our branching rametes, as 2 did produce a panicle, 29 did not, and with our dead rametes, 31 out of 31 did not produce a panicle. So just in our first year alone, we're finding that seed production is really limited in these moth-damaged plants. So overall, only 3% of our damaged rametes actually flowered. And the last most surprising result, because we didn't actually set out to, monitor this, it's really hard to find in the field, but we actually found that 8% of our damaged rametes had this pupal casing. So you'll see this here, but this is just evidence that the pupa moth had actually emerged as an adult moth. And so that's really hopeful in terms of next year's monitoring. Like Michael said, in second year monitoring, we found damage. So, we'll be returning in 2023 to keep monitoring this. And hopefully they found a mate and were able to reproduce. And so for our rametes, with our first year releases, they show this immediate response to damage. The larval feeding reduced stem height and the biomass of the stem, the leaf, and the whole ramete. Branching, even though it's trying to compensate, it's not able to compensate for the biomass losses. And flowering and seed production is very limited in our moth-damaged rametes. Moving on to my next research question, I'm interested in, at the plot level, how is plant growth and reproduction impacted by larval feeding from the biocontrol agents? And this question sounds really similar to my first one, but I'm looking at a different scale now. So when we're looking at individual rametes, it's just one ramete. At the plot level, it's a collection of rametes in a given area. And so to address this, we kind of did 2 things. The first one was these plot level measurements using 1 meter squared plots, and we were recording the damage density. And Michael's already talked about that, so I won't get into that too much, but we did see damage at our sites. So we found about, on average, 20% of stems in a 1 meter squared area were moth damaged. And then we also measured the flowering density and canopy light interception. So the canopy light interception is really important for Phragmites, and it shades out a lot of light. So we measure the light intensity above the Phragmites, above the canopy, and below. So what percent of that light is actually reaching the ground. And the second thing we're interested in is this plot level biomass. So we cut all the stems above the ground for Phragmites above ground biomass using a bit smaller quadrats because it was a lot of work. So we used 4 meter squared quadrats, and we measured the stem height prior to cutting. So we get stem heights in this first year of response to our localized damage. And when we look at the plot level stem height and biomass, we find reductions that are pretty consistent with our ramete level results. So looking at mean stem height, here's the control plots, and we're just comparing to our moth release plots right now. And we find an 18% reduction in these moth release plots of stem height relative to the control. But you'll notice that this is still over 2 meters, but just like how we saw our damaged rametes were much shorter, on average, the stems and the release plots are much shorter as well. Moving to our plot biomass on the right, when we look at our control plots as well as our moth release plots, we find that there's about a 34% reduction in above ground biomass. And so this is because we've released them at these certain points, and the damage is really centered around those points. And so we're starting to capture some of that. Just like how our ramets responded with reduced above ground biomass, our plots, we see a similar effect. When we move on to flowering density and canopy light interception, it gets a little bit more complicated. So with flowering density, you'll just notice the size of the error bars here. They're pretty large, and that's because with our control sites, even without any moth release, we saw this huge variation in the data. So some of them had 0% of ramets that it flowered, and some of them had up to 90%. And so when we add moth release, we're not really seeing any effects now, but with biocontrol, it might still be too early to detect any differences. With canopy light interception, it's also very interesting. If you look at the axes here, these values are both above 98%. So that means less than 2% of light is actually hitting the ground. And so, like I mentioned, the stem height was reduced, but it's still above, on average, 2 meters tall. And so that's still quite taller than our native plant species, like our cattails, as well as all the different sedges and stuff. So we're not seeing really the effects of moth release yet on canopy light interception, but our moth releases are quite localized, and we have space between all of them, so we can compare all these different types of releases that Michael was mentioning. So what's around these plots is kind of a wall of intact or healthy ramets. So with our plot level responses with biological control, we expect that they'll take a little bit more time to be evident, but the damage that we found so far is localized to our release sites, and we found reductions in stem height and plot level biomass. And the light is intercepted by these, dense canopy of Phragmites that's still intact. And I'll move on to my last research question. So I'm interested in how does larval feeding promote plant community recovery? And so within the first year, we're monitoring this, and we're going to be returning in later years to keep monitoring these sites. But it's a little too early, so I won't present any results, but I want to share what kind of monitoring we're doing. So we recorded the richness and abundance of co-occurring plant species, and from this, we can get a variety of metrics. So we can get the plant species richness, native plant species richness, a floristic quality index, which is a calculated value which tells us the quality, or like it's a number, tells us the quality of flora within our plots, as well as non-target plant abundance. So if Phragmites is our target plant, we're interested in how the rest of the plant community changes in response to biocontrol programs. And so we'll be monitoring this for at least a period of 5 years post-release. So our first release took place in 2022, and we'll keep monitoring that. So in conclusion, we saw these really great evidence of damage on the individual ramets, and some of that translated to our plot level measurements like mean stem height and plot biomass. And overall, we're pretty encouraged by the results. The results are promising for plant community recovery. It's still quite early on to tell, but we do remain hopeful. And over time, we expect that the moth populations will build up at our patches, and they'll be able to spread evenly throughout. So with that, I just want to thank some of our research partners, collaborators, and volunteers. We have the whole biocontrol team and all the partners on that project as well as the Waterloo Wetland Lab. I'll just give a shout out because 2 of them are talking right after me. And then Rare Charitable Research Reserve and Ministry of Natural Resources and Forestry for the funding, as well as want to thank the Ontario Phragmites Working Group for having me today. So I hope there's time for questions, but if not, I'll stick around in the chat as well.
Thank you very much, Claire. That was excellent. Always great to see the great work going on. It looks like we still, unfortunately, have some of the questions from Michael's mixed in, so I'm going to try and piece out which one is yours. As you increase your program, are you seeking out more sites in Southern Ontario?
I think it would be great to keep monitoring the different responses to biocontrol at our different sites. So that's something we're, hopeful about, and we'd certainly be interested in, but have to coordinate all the resources and everything for that.
Definitely. And I have a question for you. You had mentioned that you didn't quite see that Phragmites was still higher than, say, cattails and other species like that, and that's because it was still fairly local, the release of moths. Are you expecting, as there's sort of a greater spread, I guess, or dispersal of the moths, that you would see a change in that height?
Yeah, it's hard to tell right now. Certainly, we hope that's what we see. But in terms of what we're seeing, our where we release the moth is really localized to just a couple points because we have to prepare all the releases. So it's easy to put them just at a couple points throughout the patches. But these moths will hopefully spread throughout the patches as the adults lay their eggs. So that's something hopefully we'd want to keep monitoring in coming years.
Great. Well, hopefully we do see that. That would be great. I think just for the sake of time, we're going to move on to the next speaker, but I know there are some questions in here, so I'm sure Vicki will put them in the chat for you. If you can answer them there, Claire, that would be excellent. So thank you again for your presentation.
Thank you.
Excellent. All right, and I will invite